Okay, good evening. Welcome, everyone. Tēnā koutou, tēnā koutou, tēnā koutou katoa. That is the traditional greeting in Te Reo Māori, which is the language of Aotearoa, New Zealand, which is where I am greeting you from this evening. So I can also say good evening or good morning or good day to you wherever you are in the world and wherever you are dialing in from today. It is my great pleasure to welcome you all to this evening's Virtual Physiological Human Institute Keynote Webinar Series. Just a quick note about this seminar series before we jump right into it. This webinar series is organized by the Virtual Physiological Human Institute Student Committee. It's a quarterly seminar series, and it's a forum for us as student committee members to access the senior scientists within our in silico modeling community and get in touch with their expertise, and also hopefully provide something that is of value to the VPH community as a whole. So as you may or may not know, the VPH Student Committee has a 2-year term, and that is currently coming to an end at the end of this year. So the call for new VPH Student Committee members is currently open. So if anyone is listening and is keen, please do consider joining the VPH Student Committee. It has been a great experience. So moving swiftly along, it is my great pleasure this evening to introduce our speaker. Alys Clark is an associate professor at the Auckland Bioengineering Institute here at the University of Auckland in New Zealand. Alys obtained her degree in mathematical sciences from the University of Oxford, and her master's and PhD degrees also both in applied mathematics from the University of Adelaide in Australia. We have been very lucky to have Alys with us at the Auckland Bioengineering Institute since 2008, a postdoctoral research fellow in the lung research group, and she is currently one of the lead researchers in that group. She is a past awardee of the Royal Society of New Zealand's Rutherford Discovery Fellowship and has also received the J.H. Mitchell Medal for Applied Mathematics. Here at the University of Auckland, she was also part of a team that was awarded a Research Excellence Medal in 2018. At the moment, Alys leads research at the ABI's Reproductive Health and Pregnancy Health Group, which uses computational modeling to improve reproductive and pregnancy health. Thank you so much for joining us this evening for this webinar, Alys, and I'll hand it over to you. Oh, I should say some quick housekeeping, sorry. If you have any questions at any time, please feel free to pop them into the question box and we will address them at the end. Otherwise, at the end of the webinar, we will also give you the opportunity to raise your hand and you'll be able to unmute yourself and ask your question directly.
Thanks, Robin. Kia ora, hello, and thank you for coming to listen to my talk today. So today I'm going to tell you about the work that we're doing on modeling of maternal and fetal interactions at the placenta to help to understand pregnancy health. And just on this front slide, I have a picture of really my main collaborator in this work, Associate Professor Joe James. So whenever I'm talking about experimental work or cell biology or tissue imaging, it's really Joe who has led that work, and throughout the talk I'll talk about the other people that have contributed to different aspects of this work. So I'm interested in pregnancy, and the main thing that I'm interested in with respect to pregnancy is a condition called fetal growth restriction. So fetal growth restriction is where a baby fetus doesn't grow as well as it should do. So what that might look like in a pregnancy is that over the course of a pregnancy, the size of a fetus will be estimated. We're expecting the size of that fetus to grow along a normal growth curve, but at some point that growth drops off and slows down. And so that baby isn't growing as well as it should. It's not the same as a baby being small, but growing normally along the curve. It's an abnormal growth of that baby. So why are we interested in that condition? Well, it's one of the leading causes of stillbirth, so it's important in any pregnancy to try to identify any problems that might increase risk of stillbirth so that those pregnancies can be monitored. Babies who are grown from pregnancies that have fetal growth restriction are often at higher risk of health complications later in life, things like cardiovascular disease or diabetes. So it really is a condition that impacts that baby throughout their life. Now, there's quite a few problems with this condition. The first of all is we don't really know exactly why FGR occurs. It's quite a complicated condition. And because we don't know why it occurs, it's very difficult to actually predict which pregnancies will go on to develop FGR. So if we're wanting to predict at this early stage in pregnancy whether the baby is going to fall off that growth curve, that's very difficult. And because of that, actually, clinically, up to half of pregnancies where babies are smaller than they should be, those babies are born and that condition hasn't been detected during the pregnancy. And we know that just monitoring those pregnancies really improves outcomes. So detection is really critically important. And if we can't predict those pregnancies, if we don't really have a great understanding of why it's happening, how can we expect to treat that condition? And at the moment, there isn't really an effective treatment for pregnancies that have fetal growth restriction. So one thing that we do know is one of the leading causes of fetal growth restriction is something called placental insufficiency, but that in itself is a little bit complicated. So the placenta forms, it's a fetal organ, and it forms in the uterus, and it attaches to the wall of the uterus. So one reason that a pregnancy might suffer from fetal growth restriction is because of a reduced blood flow to the placenta from the uterus. So that's an issue with maternal blood flow to the placenta. It might happen because of the formation of the placenta itself. And as I mentioned, that's a fetal organ. We're expecting the fetal organ to develop alongside the fetus. And if that doesn't happen quite as it should do, then the baby might not get the nutrients that it needs to grow as it should. And finally, there might be an issue at the interface. So where the maternal circulation, the blood flow full of nutrient-rich blood from the uterus goes over the surface of that fetal organ, the placenta, and delivers oxygen to the baby. If that interface doesn't develop as it should, then potentially the delivery of oxygen and nutrients to the fetus won't be adequate. So the placenta develops on demand to provide oxygen and nutrients to the fetus, and it really is quite a complex structure because it has to provide up to around 16 meters squared of surface area for effective exchange. So just on the top of this slide here, we have some pictures of the placenta at different scales. It has a really complex vasculature. There's blood vessels that form over the surface of the placenta, large kind of highway blood vessels, but the tissue itself is very delicate and intricate. And there's a great scale difference from those large blood vessels that feed the placenta and the tiny little capillaries that participate in exchange. And really, like if we think of the adult exchange organ, the lung, we're seeing a similar level of complexity in order to provide that large surface area for exchange. But unfortunately, when we're doing diagnoses of what's going on in the placenta, we rely quite heavily on ultrasound, which is really quite a low resolution technology, and we really can't see that intricacy in structure. And so abnormal structures are often not spotted until the baby is born. Because stillbirth and fetal growth restriction is a big deal, there tends to be a lot of different types of research that go into trying to improve outcomes in pregnancy. There's a lot of research in pregnancy on the genetic risk factors for pathologies, including fetal growth restriction, studies on cell behavior, cell organization, tissue structure and function, and trying to relate that to outcomes, right up to those kind of in vivo clinical studies. And so computational modeling, which is where I focus on in pregnancy, really has a great role to play in trying to join together some of those different measurement techniques that are used to investigate pregnancy and try to understand a little bit more, add a little bit more to what's understood about conditions of pregnancy like fetal growth restriction. So this slide really summarizes what my group is aiming to do. We're looking at all the time trying to find new ways to measure structure and function in pregnancy. So that might include ways to measure what the anatomy of pregnancy looks like and of the placenta, trying to bring new insights into how cells behave in the placenta and bring that anatomy and cell behavior together, new tools to analyze imaging data in pregnancy, and ultimately new ways to clinically image blood flow. We bring that all together via a virtual pregnancy. Today I'm going to talk about those three different areas of a pregnancy that might contribute to fetal growth restriction: the uterus, the placenta, and the interface between them. So I'm just going to tell a few short stories about the work that we're doing in order to try to paint a picture of the intricacies of pregnancy, but also the scope for computational modeling in this area. So I'll start off by talking about the uterus. So I've mentioned before that the placenta is basically embedded in the uterine wall. So the non-pregnant uterus has quite a complex network of blood vessels that lie through it. It has large arteries that feed the uterus and then arteries that feed the tissue. There's some tiny little arteries called spiral arteries, which sit actually in the tissue that is shed during menstruation. And these arteries play a really important role in pregnancy. When the placenta, when the embryo implants in the uterine wall and the placenta starts to develop, placental cells actually invade into the tissue of the uterus, and those tight coiling arteries, the spiral arteries, are transformed into really quite large funnel-like arteries. And then there's a whole network of blood vessels within the uterus that also remodels and grows to accommodate a 15-fold increase in blood flow in pregnancy. And in ultrasound, it's the largest artery that is measured. It's really the only artery that is accessible to ultrasound measurement. But of course, there's a lot more going on downstream of that. And the fetus itself lies right down through the placenta, through the umbilical cord, and quite far away from the site of measurements. Although there are ultrasound measurements of the fetus are important as well. So that process I was talking about, how the blood vessels of the uterus change in pregnancy, it's really quite an amazing thing. So I mentioned that there's spiral arteries in the tissue of the uterus, and they really are very small, tortuous, spiraling arteries. And in the early stages of pregnancy, placental cells actually invade from the placenta into the tissue of the uterus, and they initially form plugs in those spiral arteries, so little plugs of cells that prevent too much blood getting to the placenta in those early stages of development. And importantly, they prevent red blood cells getting to the placenta. And that's quite important because actually a lot of the developmental processes in the early stages of development rely on a low oxygen environment. And then later on in development, those plugs disperse, and actually the muscle in those artery walls is replaced by placental cells, and that allows for a large amount of blood then with high oxygen to circulate around the placental structure over the surface of the placenta, and the blood vessels in the placenta itself can then take up that oxygen and nutrients and prepare for healthy development. So that happens to the arteries that are really close to the placenta, and that's really dramatic. And so those arteries have been really studied quite heavily over the years. But every other artery in the uterus actually changes its form and function, partly in response to hormones and growth factors, which tend to relax the vessels. They get larger, but also due to that large increase in blood flow that I talked about. And then there's these quite mysterious connections that are shown in my picture by little blue, red to blue connections, and they're arteriovenous anastomoses. So they're direct connections between the arteries and the veins in the uterus. And they're thought to be protective of high blood pressure or changes in blood pressure, so that the placenta doesn't experience sudden changes in the hemodynamics or the delivery of blood to its surface. But they were only really discovered in around about 2005, so they're quite a recent discovery in anatomical terms. What we're measuring in pregnancy in the looking at the uterus is what's called an uterine artery Doppler. And so what we're looking at is velocity in the biggest uterine arteries, and we see these peaks in velocity and then a flattening, a plateau of the velocity in a healthy late gestation pregnancy. In an abnormal pregnancy or early in pregnancy, we see an increase in blood flow velocity and then what's called a notch. So a second peak in the velocity over a maternal heartbeat. And one of the great mysteries of pregnancy is what exactly causes that notch. It's seen in fetal growth restriction, it's seen in other conditions such as preeclampsia. It's generally a sign that the arteries downstream of the uterine artery are too high resistance. But what are the contributors to that? Sorry, I expected some text to come up on this slide, but I'll just have to tell you about it. So our interpretation of that Doppler imaging actually comes from a computational model of the uterine circulation that was developed in the 1980s. And in that model was the wave transmission model. There was an input flow waveform, so a flow of blood arrived from the heart through the circulation to the uterine artery. And then the blood travels down the uterine artery to the insemination site. And the whole of that complex vasculature of the uterus was represented by a single load impedance, which is assumed to be dominated by those really tight coiling spiral arteries. And so since the 80s, the interpretation of that Doppler waveform has been that it's really dominated by those spiral arteries. So we wondered, now we know a little bit more about the structure of the arteries in the uterus, and in particular, those connections between the arteries and veins, whether a more anatomical model of that circulation might tell us a little bit more about what is driving that Doppler waveform, because it's really not a great indicator of pathology. And what we saw when we took a similar transmission line model, but a more accurate representation of the uterine vasculature, is those anastomoses, which had been hypothesized to protect against high pressure events, actually also limit the detection of problems downstream in those spiral arteries. So if the resistance of the spiral arteries remains high through pregnancy, but there's an established anastomosis network, you simply won't see a notch in a spiral artery, even if the placentation is not great. So that led us to think, well, what does drive the notching that we're seeing in abnormal Doppler ultrasound? And what we then did is we looked at the arteries upstream of those anastomoses, which starts quite small in pregnancy, of a similar size to the spiral arteries, but they only they don't have that really dramatic remodeling. They do double in size, but they don't go from hundreds of microns to millimeters like the spiral arteries do. And we found that actually just by perturbing the radius of these larger arteries called radial arteries over pregnancy, we could replicate notching in the Doppler waveform. And that notching occurred at roughly the radial artery radius that you might see in a pathological pregnancy. So that more anatomical model was suggesting to us that there's a little bit more going on in the uterus in pregnancy, what has been interpreted to be and arteries in the system matter as much as some of those spiral arteries, those dramatically remodeled arteries. So that modeling really showed us that the trophoblast independence, so that the remodeling that is not because of the invasion of those placental cells into the uterus, and that more traditional type of response to an increased blood flow in pregnancy, that remodeling in those upstream arteries might be just as important, at least to interpreting Doppler, but it's really not been studied well at all. So that set us off on a path of trying to understand a little bit more about the uterus in pregnancy. How does the anatomy of the uterus develop over a pregnancy? What are the consequences of that anatomy for blood flow? And how do uterine blood vessels actually functionally respond to pressure and flow? And if we can understand that, then hopefully we can develop a multi-scale model of the entire uterine circulation to try to understand the system rather than the individual arteries that are contributing to that system. Now, understanding human anatomy in pregnancy is actually quite a difficult thing. It's very unusual to observe a human uterus, a pregnant human uterus. And so to understand human anatomy, we actually had to go to historic collections housed at the University of Cambridge and look at individual slides of pregnant uteruses that were collected kind of in stacks. So individual slices stained in different colors through the uterus. But using some image processing techniques, we were able to account for all of those different stainings in those uterine slides and actually align the tissue slides to show what the tissue structure looks like in 3D through uteruses, pregnant uteruses. And when we looked at the arterial sizes in those blood vessels, those radial arteries that we think are important to the Doppler signal, they increased in size to roughly double their size at around mid-pregnancy. And then the spiral arteries, which are the ones that were dramatically remodeled by placental cells, they increase right up into the second trimester, and they become actually quite a lot bigger than radial arteries. So if you think about the arteries of the uterus, which are feeding the placenta, in much in the same way as your garden hose is feeding your herbs or your plants in your garden, the spiral arteries, which are a big kind of funnel artery at the front, are essentially the sprinkler. They're providing blood to the placenta at a low velocity that doesn't damage the placenta. But it's the hose that is responsible, or the radial arteries, those upstream arteries, that are responsible for how much blood actually gets there. So the volume of blood. We can't do everything in humans when we're trying to understand the uterus and the placenta. So we're also working on rat models of the placenta and trying to understand what the arterial structure of the rat is and comparing structure and function in rat to humans. So these images show micro CT images of non-pregnant and pregnant uteruses, showing the same types of vessels as we're seeing in the human. The pregnant side here in yellow, we see the larger uterine arteries coming through to the radial arteries and spiral arteries, and then feeding the placenta. With this information on anatomy, using relatively simple transmission line models, we're able to then predict what Doppler ultrasound will look like in response to both the normal anatomic structure and the perturbed structures that might be reflective of pathology, both in rodent models and in human models, taking into account their differences in anatomy. With humans, we have those anastomoses that I talked about quite far down our arterial structure, and in rodents that sits higher up. But there are a lot of actual similarities between rodents and humans. We're also able to then predict how the differences in anatomical structure might actually impact the function of those arteries in terms of blood flow and hemodynamics. The next thing we're able to do in rodents that we're not able to do in humans is we're able to isolate arteries and look at the response to things like pressure and flow. So in this study, we took isolated rodent arteries, pregnant and non-pregnant, and we looked at pressure responses in those arteries where we turned off the active response. So there's no calcium, and so the smooth muscle in the arteries can't contract. And what we found was pregnant arteries have a higher compliance, so they're less rigid than non-pregnant arteries. They have a much larger diameter, and all of that contributes to a steady perfusion of the placenta. The next thing that we did was we allowed for the smooth muscle to function. We looked at the responses to pressure and flow. And what was interesting when we did that, so the smooth muscle that acts in the vessel walls actually predominantly in pregnant arteries doesn't really respond that much to flow at all. So if you increase the driving pressure for flow or flow through a pregnant radial artery, you see that the smooth muscle doesn't really respond that much until you get quite a lot of flow. And that actually the non-pregnant arteries are more sensitive to flow. So if an artery didn't remodel as it should, if the hormonal environment wasn't as it should, then potentially you might see an oversensitivity to flow. What we can also do in these rodent models is we can take non-pregnant arteries and we can add paracrine factors. So we can add hormones to our experiments, and we can see whether we can make non-pregnant arteries behave a little bit more like pregnant arteries. And so the question we were trying to answer there is what actually makes radial arteries react pregnant? So we looked at estrogen, placental growth factor, VEGF, and as well as progesterone, and a combination of all of those factors. And we found that certain hormones, such as estrogen, could help to allow a non-pregnant artery to act a little bit more like a pregnant artery. So we're diving down a little bit more into those hormonal mechanisms, that unique hormonal environment of the uterus, and how that affects how arteries function. So where are we heading with all of this? Well, with a computational model of vascular reactivity in an individual vessel that responds both to hormones but also to increases in blood flow reflective of pregnancy, alongside data in rodents, which allows us to kind of separate out those effects of pressure, flow, hormones, and then vascular models at the network scale, which we've developed for both rodents and humans. We're hoping to be able to perturb the response of vessels within that network and the response of the network to different flows and hormonal conditions, to see actually what happens if the conditions are not right for vascular remodeling. And then ultimately to scale that up to ultrasound to understand which vessels actually matter the most to both a pathological pregnancy, but also what can we detect in ultrasound. The next short story I'm going to skip on to is around the placental or the fetal circulation. So the placenta is actually delivered after birth. So compared to the uterus, it's a lot more readily assessed. The placenta in fetal growth restriction is normally thinner. It's normally smaller than a placenta from a normally grown pregnancy. The vascular branches are shorter, and there's fewer vascular branches by up to 36%. So in this image of a normal placenta, you see quite regular vascular branching. In growth restriction, the placenta has much longer, thinner branches and really not that kind of regular vascular branching pattern that you might see in a typical vascularized organ. So what we're able to do is to take data really in any form that we can acquire in pregnancy, from photographs of the delivered placenta, ultrasound measurements of what the vascular structure looks like in vivo, to probably the gold standard, which is micro-CT imaging of the placenta. We're able to extract vascular networks from that placenta and use electrical circuit analogs or wave transmission models to predict both the resistance of the system, the distribution of blood flow through that anatomical system, and importantly, how does that vascular branching geometry relate to clinical measurements like ultrasound, in this case ultrasound in the main umbilical vessels that connect the placenta to the fetus. We can do this on the population level by statistically matching branching geometries to morphometry measured in particular populations. So we can reflect that normal placenta and the thinner, smaller, less vascularized placenta of FGR, and predict the impact of typical FGR parameters. For example, in severe FGR, our model predicts a 7 to 8-fold increase in placental resistance and decreased perfusion through the placenta. We can also look at patient-specific models. So think about when the placenta is delivered and we're able to micro-CT that placenta, what just looks unusual and what is actually a bad outcome for that placenta. And I'll cover off some of these shortly. So in our models of FGR placentas, I've mentioned that we predict this increase in resistance, but interestingly, we also predict increased capillary shear stress, because even though the tissue of the placenta receives a lower blood flow, there's significantly less capillaries for that blood to get to. So we predict in that capillary level where the placenta is developing an increased capillary shear stress. And now that's interesting for the pathogenesis of fetal growth restriction, because in these population-based models, we're predicting a lower perfusion but an increased stress on the tissue. We went back into the lab and we looked at cells migrating under the shear stresses that we predicted in fetal growth restricted placentas, and we saw that those cells moved more slowly and they moved more persistently, which means that instead of randomly migrating, as you might expect cells in a culture, they moved in the direction of flow when we were exposing them to fetal growth restricted levels of shear stress. We know that persistent cell movement in a vascular capillary actually inhibits vascular branching. It makes it harder for branches to form. And so that kind of leads to the question of whether that abnormal vascular structure in growth restriction might be established relatively early in pregnancy, and the lack of vascular branching that we're seeing in delivered growth restricted placentas are actually something that emerges from an initial problem in growth, which then feeds back to higher shear stress, increased persistence of cell movement, and less branching. So that's a question that we're still looking into. In patient-specific models, we can start to think about questions of placental vulnerability. Here are two placentas which have been imaged in London by micro-CT. This is quite a symmetric placenta with a central cord insertion. This one has a cord insertion off to the side. But what was really interesting in this placenta too is that not only did it have a more asymmetric branching structure, it also had small vessels that feed the placenta that were quite constricted compared to others. And we saw this patchiness in placental function. And what was interesting about this is in terms of the resistance of the placenta, while both had normal resistances in our models, when we started to kind of add insults on top of that placenta, placenta 2 was a lot less robust to insults. And that suggests that maybe the heterogeneity in branching structure or patchiness in placental perfusion might be important to placental health. And that's important because we can actually measure some of these quantities in MRI. So we have ongoing work trying to understand how that patchiness in function or in structure might look in terms of function in a growth-restricted placenta where we expect a more heterogeneous structure. Moving beyond fetal growth restriction, another application of these placental models is in understanding a condition known as twin-twin transfusion syndrome. In twin-twin transfusion syndrome, you have two twins that share a placenta. And what can happen is that one twin can dominate the other. It can take most of the flow from the placenta and it grows a lot better than the other twin. And you're able to do surgeries on these placentas, basically to sever connections between the two twins back to anastomoses, so connections between the placentas of the two twins. But which are the important connections to remove in a surgery? So if you could imagine that in a twin transfusion syndrome surgery, that you could map the surface of the placenta and the vessels on the surface of the placenta, which is quite feasible with technologies that are evolving in imaging. If we can map those blood vessels to a computational model, a personalized computational model, and we could also map where there are connections between the two placentas. So we can see arteriovenous anastomoses, venous anastomoses, and arterio anastomoses. So connections between actually the two twins' blood vessels, direct connections, which can contribute to transfusion from one fetus to the other. And with our models, we're able to identify, in this case, the arteriovenous connections, which are leading to that transfusion of blood, which can be targets for surgery. So these models potentially could aid decision making in those cases. And in terms of imaging the placenta, I showed you some whole organ micro-CT images. This image is a micro-CT image at a high resolution of a placental cotyledon showing, so that is a functional unit of the placenta, showing really the complexity of branching structures in that organ. So all of these bright lines are blood vessels. And you can almost see large highway blood vessels that go deep down into the placental tissue, and then this really intricate network of blood vessels. And these structures we want to be able to represent in models, but obviously network models and transmission line models are just going to be too complex to describe this. So we're looking for homogenization approaches that could try to give us a balance between understanding the complexity in that vascular structure and computational tractability in models. So finally, I'm just going to quickly talk about the interface, that barrier between the placenta and mother. So the placenta itself is fed by the umbilical cord, and the fetal blood vessels sit within what are called villus trees. We have the spiral arteries that I talked about right at the start of the talk, which feed blood out into that placenta. surface, and we think that the interaction between the maternal blood and the developing placenta might influence its growth and its function. So one of the more recent suggested indicators of placental health in terms of ultrasound is to look at blood coming from spiral arteries in what is called jets into the space that surrounds the placenta, the intervillous space, the maternal blood space. And so we had a look at a large number of images of these jets of blood, whose length have been associated with conditions such as fetal growth restriction. And we also looked at a bunch of images of the placenta and looked at the tissue density in those images. And using quite a simple model representing the opening of a spiral artery and a porous medium representation of the placenta, we were able to show that we simply could not have blood penetrating the depth that these jets do from spiral arteries if the tissue structure really was as dense as what is seen throughout the whole placenta. And it suggested that there must be empty spaces forming around those spiral artery openings. So almost like what you saw in the placental image, where there was a highway for blood to get into the placenta, there might be highways for blood to get from the maternal side as well. And so what these highways are called, these empty spaces, are central cavities. So they're cavities of tissue-free region at the entrance to the spiral artery openings. And we've predicted, using quite simple models of the structure of the placenta and the relationship between structure, blood flow, and oxygenation, that they might be fundamental for oxygen exchange. So if blood comes out of an artery and it hits a bunch of tissue, blood can't penetrate very deep. It's the oxygen-rich blood from the mother. And so if there's an opening in which a jet can penetrate, that will help oxygen to make it all the way through the placenta. We've also shown that these jets, or these openings, must grow progressively over gestation. So such a relationship requires, in some way, that the placenta forms around the spiral arteries, so it senses and responds to this flow coming from spiral arteries. So again, we went back into the lab and we looked at whether there was perhaps a potential for the surface of the placenta to sense blood flow, and indeed there is. The cell, the syncytial trophoblast that covers the entire placenta, actually expresses a number of shear stress sensing proteins, and it does so much more strongly in the first trimester than at term. So while that placenta is developing, it has more capacity potentially for sensing shear or flow from those spiral arteries. If we look at the anatomy of the placenta in the first trimester versus term, and we simulate using porous medium models the flow through that placental structure, and also we can do explicit simulations on little blocks of tissue that make up that placental structure. So we can parameterize tissue block, which are not the size of the placenta, but we can look at little subsections explicitly, and we can see that actually in the first trimester there is a relatively higher shear stress than at term. So the placenta is actually feeling a higher shear stress, and has more capacity to respond to that shear stress. So the next question facing us, where we're going with this, is how does that actually influence growth and development of the placenta? And is it possible that abnormalities in structure early on in pregnancy, like we saw in the fetal growth restriction placental circulation models, actually have an influence on placental development in the same way, but this time from the maternal side? So just to summarize, there's a lot going on in pregnancy. The placental circulations, both on the uterine side and the fetal side, are dynamic. They change a lot during pregnancy, and that makes them quite difficult to visualize clinically and to model and to investigate in the lab. But hopefully we're starting to bring together some of the data that describes these circulations via computational modeling. And I think the most important thing that is coming out of our modeling is that nothing in the system happens in isolation. We need to think of the uterus, the placenta, and the fetus as an integrated system. We believe that understanding how changes in anatomy affect function of that system really allows us to better understand what is driving the exchange capacity of the placental, and that's ultimately going to be what drives fetal health and fetal growth. And we hope that bringing all of this information together will ultimately allow us to develop new ways to look at the baby and to detect at-risk pregnancies. So bringing together a whole bunch of structural metrics that we gain from micro-CT imaging and other imaging such as ultrasound or MRI, model predictions of function based on that structure and the interactions between structure and function as the organ develops will hopefully bring us new insights into exchange capacity and fetal health. And I'll just end with thanking all of our funders, all of our current and past group members who've contributed to this work, and a bunch of other collaborators conducting many different modalities of imaging or investigations that have contributed in some way to the work that I've presented today. And thank you everybody for your time.
That's fantastic. What a great talk. Thank you so much, Alice. I'll open up the floor to everyone now. If you would like to ask a question, feel free to type your question into the question box, or you can also raise your hand, and I think you should be able to unmute yourself and ask a question. I might kick off the questions, and yeah, actually your talk really anticipated the question that I was going to ask, which is really to do with the translational aspects of your work. And you know, the VPH is really interested in the idea of the clinical digital twin. So I'm always interested in what does that actually look like? What is the pathway to translation? How is this actually going to get into the hospital? But I thought your example of twin-to-twin transfusion syndrome was just such a beautiful example for how you actually bring computational modeling into the clinic. And I think the next question then is, how do you actually show that it improves outcomes for patients? So do you think you're still very far off from a clinical trial to see if we can bring computational modeling into the clinic?
Yeah, I think that there's ways to bring the models into the clinic more quickly, but maybe in a simplified manner, and then the longer term goals. So one of the key things that we can do is to take some of the learnings that we've got from our models of Doppler ultrasound and which arteries might matter in that context, and suggest new features in Doppler to look at, which you can look at large cohort data and that has been collected over a bunch of kind of different studies, and that's a lot simpler. I also think there's some potential for our anatomic studies to be used to guide development of new imaging techniques, because a lot of the new ultrasound techniques are trying to capture more of that downstream vasculature, but we don't really know what it looks like yet. So I can see interactions with models there. And then that actually building in models to clinical ultrasound, I think, will be a hurdle, because it's typically quite hard, as everybody knows, to get into any clinical process, but in pregnancy the hurdles are greater. But I think there's a keenness in the community to have modelers involved, so I think that's a positive step.
Yeah, I mean, yeah, there's definitely a keenness to improve outcomes. So yeah, if models can be that mechanism, then yeah, it's definitely very, very welcome in the clinical realm. Great. I see we have a couple of questions now, so I might just go through the questions and read them out. I'll start with Irene, or Irene maybe, who thanks you for your great talk. The first question is about which MRI technique is used for perfusion heterogeneity.
So most of the MRI techniques that are used in the placenta are based on brain imaging. The most common technique for perfusion would be an IVIM, and diffusion weighted image, and an assumption of a kind of quasi random distribution of blood vessels in a intravoxal coherent motion model. The model that we use in Auckland comes out of UCL and King's College, London, and it's designed to try to separate out the maternal and the fetal components, but it's essentially a diffusion weighted imaging. And then T2 imaging is often used to look at oxygenation in some way or form. So hopefully that answers that question. And then
yeah, Erin's second question was about how do you explain the difference between a more regular versus a more irregular vascular network in the placental examples that have the same resistance? Just with a note that in their experience, heterogeneous trees lead to higher resistances.
Yep. So the heterogeneous tree in that case, we did predict a slightly higher resistance, but still in the normal range. So not exactly the same resistances. There's also, because those are patient-specific models, they don't have exactly the same volumes, overall vascular density, so that other tree had some good regions and some bad regions, but it was more resistive. It just wasn't abnormally more resistive, I guess. So I think that you're right. And in a one-for-one comparison between a more regular and more irregular network, we would see a higher resistance in the more irregular network.
We also have a question from Banu, who asks, how do you distinguish between the arterial and the venous structures on micro-CT images? And is it due to the standard anatomy predictions, or is there something in the imaging that's giving you that information?
Yep. Different ways. So usually the arteries and veins come paired, so there's reasonably a one-to-one correspondence, and the veins are generally bigger than the arteries. Also, the connectivity between back through the tree. So we always, whether or not we're imaging, we don't in Auckland image the whole placenta. The whole placenta images have been done in London, but in Auckland, where we image just parts of the placenta, we always take photos of the placenta before we do that micro CT imaging, and you can quite clearly see the difference between arteries and veins there because of where they lie on the surface of the placenta, and the connectivity back to the umbilical cord, because there's usually two umbilical arteries and one umbilical vein. So we match them back to those photographs, and then we can and then we can separate them out by their connectivity and their size. But on the photos or the images that you would just take of the surface of the placenta, which attaches to the umbilical cord, there's an anatomical difference between the arteries and veins, so you know which ones are which.
Fantastic, thanks. And then another question about how long it takes to finish the study. I'm not quite sure if that might be referring to the imaging part of things.
Yeah, so the time taken to construct a model, I guess. Do we have a clarification on that, or?
I'm not entirely sure, Banu. Feel free to expand on that if you want.
I can tell you that it takes a lot less time than it used to put these models together and to run them. And I have, I think, reasonable confidence that we can. So in the very large placental networks that we simulate, where we have explicit representation of many thousands of arteries, we can solve those models on desktop computers in, 5 or so minutes. So it's not a terrible computational constraint. And I have reasonable confidence to simplify those down, but we haven't yet started the process of trying to take those more complex anatomical models and still make good predictions, but with simplified models. But that's definitely in our future, because obviously real time is going to be quite important for clinical use.
Yeah, absolutely. Oh, fantastic. Thank you so much, Alice. I think that's pretty much it for our time this evening. I'd like to thank you once again for an excellent presentation, a really, really interesting, really exciting field. Yeah, really, really pleased you could join us for this webinar this evening. Also, big thank you to everyone who attended. And with that, I'll say thank you and goodbye to everyone.